while the slides are getting there, folks, thanks very much for the opportunity to be able to share some of the things that we're doing in pancreatic cancer. Now, we know pancreatic cancer is a difficult disease to treat. We have more and more treatment options accumulating, but unfortunately they only work in a very, very small proportion of patients. We have some mainstays of therapy like FOLFIRINOX and Abraxane plus gemcitabine, which have given us some traction, particularly with stabilization of disease and allowing patients to be healthier and receiving other more novel therapeutics. However, historically, we've always struggled, particularly with targeted therapies, in really getting meaningful responses, including immunotherapy. So, as you know, there's a lot of genomics data and a lot of transcriptomic data now coming to light. We've contributed to some of those, and there are, as we see, it's not.
Sorry, we can't see your slides.
No slides.
The slides are coming. I'm currently downloading it.
Yes, so yeah, we'll catch up. I don't want to waste time. So we know a lot about genomic data, and we know that predominantly, apart from the four big players, the prevalence of other events, particularly those that are targetable or druggable, are small. We see some of those in the non-KRAS mutant population, and we see alternative drivers like BRAF V600E and others, and particularly fusions like ERG1 fusions, etc. But typically, we see these four mutations of KRAS, P53, P16, and SMAD4 dominating all of these things. Ah, marvelous, thank you. If we move along a few slides.
Let me know where I stop.
Next one. Perfect. So we can see that as I was mentioning, that you have these dominant mutations. We can see some patterns. We see patterns in structural variations, and we can see that this described there. Some signatures are particularly interesting, and one of the challenges is the HRD signature, the signature of BRCA-NES. And whilst we can do that in whole genome sequencing, it is still a challenge to do in the clinic. Next slide, thanks. So we can see that now we're starting to understand transcriptomic subgroups in pancreatic cancer. We see some of them associated with poorer prognosis, and predominantly two lineages that we see. Have the next slide, please. Oh. Yeah. So we see two predominant. If you look at the phylotranscriptomic tree to the right, you can see that there are two clear lineages, those that look like the pancreas, which are called classical pancreatic, and those that don't look like the pancreas and lose all endodermal identity. And they're the squamous or the basal-like or whatever you wish to call it. Some of these groups are starting to stratify into different subgroups. It's always a challenge to do that because there's differences in biology between them, and also which tools you use to separate that. But the difference between the genomic attribute of the basal-like or squamous versus the classical is mainly epigenetic. So of course we see a massive shift between the transcript of those two lineages. What is fundamentally important about this, though, is that here in the heat map on the left is a group of cell lines where you don't get any stroma, so you start to see pathways a lot clearer. And we can see that there's clear separation in not just genes, but also pathways. And I won't go into this in detail, but you see differences in metabolism. You see differences in morphogen sequence signaling, as we can see here. You can see differences in immune response. You can see differences in a whole host of different mechanisms that are critical to the cancer, but also critical to how we drug them. So it's little wonder if we look back and if we test a drug in all subtypes that we're not seeing dramatic responses. And if only your pancreatic or your classical subtype is likely to respond, then you're already diluting your signal by 50%. Next slide, please. We do see targets for existing therapeutics and some emerging therapeutics, and then at a genomic level, but we see that they occur in a very low prevalence. And so even a study of 30 patients may not detect a signal if you've got a 2% prevalent subtype. So the hypothesis being, if we better targeted those subtypes, or at least enriched them for them, we would start to see more meaningful responses. And of course, in many studies, there may be 1 or 2 responders out of 30 cases. And of course, we've seen reports of exceptional responders. So there's 2 opportunities here. One is the sort of the single genetic event, repurposing drugs that we've used in the past, or extending indications, or then understanding the transcriptome better. So in order to do this in the clinic, we set up a platform called Precision Panc. You have the next slide, please. And the concept of this platform was to take us from where we are in single trials to a platform which I think is necessary for diseases like pancreatic cancer, which are high in mortality, but still relatively only 9th or 10th in incidence. So it's difficult to do meaningful studies in single institutions at a clinical level. And moving to trial platforms, and then how we might move to real-world data in the longer term, and really starting to glean information from every patient that's treated, whether they're treated with novel therapeutics or existing therapeutics. The next slide, please. And so the goal of Precision Panc was to understand in the clinic, how do we use current treatments better? Can we define those patients that will respond to current therapies, and can we predict those that don't respond with high accuracy, so that we can we can offer them clinical trials and more novel therapies? Because if they're not going to respond, a lot of them get sick pretty quickly and don't even get a second-line therapy. We don't know much about how pancreatic cancer evolves with time and with treatment, and so if we think about precision oncology as the next way of thinking, it's not about just identifying a target and treating a patient. It's more about understanding what the patient's molecular pathology looks like in the clinical trial. How does the treatment change that? And we can see this in many other cancer types. We can see this, we understand this in breast cancer, we're understanding this in prostate cancer. What does it look like post PI3 kinase alpha therapy? Because that population of patients looks different. In pancreas, we're very much behind that, so we need to start to map out all of these things. And of course, how do we accelerate therapeutic development for pancreatic cancer? Next slide, thanks. So Precision Panc is built on 3 pillars. There's the discovery component, which is large data sets of all the genomic information, etc., that we've contributed to and others in the world have put together, which allows us to prioritize strategies, and led by Owen Sampson at the Beatson, preclinical development, where model systems are used to define a preclinical platform of evidence that takes us into clinical development, which is the primer series of studies, which I'll talk about in more detail now. And of course, information from that feeds back into the discovery program. Next slide, please. So in order for us to do this, we had to build a few things. We had to build the Glasgow Precision Oncology Laboratory, which is a lot of next generation sequencing approaches and other wet chemistry approaches as well, which was able to provide each one of these pillars with omic data. We also look after other cancers too, as well, and expanding to all solid tumours because of the technology that's available to us and the approaches that we've developed. Next slide, please. So the first thing we needed to do, we needed an affordable and fit for purpose genomic test that worked seamlessly and efficiently with the healthcare system, particularly to run Precision Panc. There is no way we could pay £3,000 per patient for a Foundation 1 test, and any of those competitors were completely priced out. Also, the timeline was too long, particularly for pancreatic cancer, so we had to do something about that. And so we built a test. Next slide, please. And this test is now called the Glasgow Cancer Test. It applies for almost all solid tumours except for some of the fusions in sarcoma and some pediatric tumours. It goes well beyond the coding regions of genes. It was designed by Philip Beer and Susie Cook, primarily on the back of curating all the databases and the literature to identify what are truly cancer genes. And it includes copy number structural variations, some genes with tile over introns, genomic signatures, certainly tumor mutation burden and microsatellite instability, and we're currently working on HRD, clinically relevant fusions, at least actionable fusions, and of course immunological resistance markers and regulatory regions. Next slide, please. And that allows us to deliver a test now to patients as part of Precision Panc. We've just moved our laboratory into the health service to streamline these activities, and each patient and their treating clinician will receive a result for their tumor profile. So what we've built in Precision Panc is the ability to biopsy most pancreatic cancer patients with a suitable enough biopsy that can be used for next generation sequencing, and we take those and we perform these diagnostics on them and return the results. And currently the success rate is well over 80%, 10% it's usually a sample issue, and then 5% it's a DNA issue. So in over 85% of patients that enter Precision Panc, they receive a test result. Next slide, please. So how Precision Panc works. Precision Panc works essentially as a master protocol with a clinical and molecular screen for patients. In this master protocol, samples are required. We perform a molecular profile. We return those results, and we also encourage patients to recruit into clinical trial, of which we have a series you can see underneath, and I'll talk about those a little bit more. Next slide, please. So the master protocol, one of the key innovations was to speak to patients very early on in their diagnostic journey, not at the time of decision making for treatment, but at the time of diagnostic workup. And so by parallel streaming both the research and the diagnostic protocol, we take the pressure off the rapid turnaround time of 5 days, although we can do that if we need to. But generally, we parallel stream that, and the molecular pathology results come back before a treatment decision is ready to be made. Next slide, please. As we build out the master protocol, we had to understand what our patient population looked like, what they required, so that we could build out an ecosystem where all patients had an opportunity on a clinical trial, whether it be directly run by Precision Panc or an affiliated study. And so we've started to populate these different indications, and if we go to the next slide, which gives you a helicopter view of what Precision Panc looks like. You can see the master protocol with a horseshoe shape. Patients come in on the left. They can enter right at the beginning, or they can enter after they've had a first-line standard of care. They could be either on one of our primer studies, an external study, or be on standard of care. And once they're in, we track them and we prepare them for second-line studies. Similarly for for locally advanced disease and also for neoadjuvant disease, trying to offer every patient with an option. And then following on from that is a second-line umbrella, which has a number of studies which are growing. Some are selected for based on molecular targets, some are selected for based on performance status, but really trying to provide patients with options at every step of the way. Could I have the next slide, please? So one of the key studies is Primer 001, and I'll dwell on this just for a few minutes. One of the challenges we have is that we don't have a biomarker for platinum response. We certainly have germline BRCA, potentially somatic BRCA, although there's no studies to clearly demonstrate that. We need to address the issue of platinum. We also have this challenge of choosing between FOLFIRINOX and Abraxane gemcitabine, which are generally equivalent therapies. Of course, that may change if there's a BRCA mutation, but BRCA germline accounts for about a quarter of HRD, somatic another quarter, and another 50% we don't have any root gene cause. And so we have a hypothesized biomarker that we're testing here. It's an adaptive study. we do regular interim analyses that allow us to define A, futility, B, whether we should change our recruitment strategy to a biomarker positive. It's a very large phase 2 study because it's powered for the biomarker, but should we receive a signal, the phase 3 completion study will be relatively small, something like 50 or 60 patients. We have the next slide, please. So where are we up to? Of course, everybody's been challenged by COVID, and we're probably around 18 months behind where we would like to be at this point in time. But at this point, we have, that should say 441 patients that have entered the study, not 341. So 441 have entered. And because we invite these patients early, some of them don't have pancreatic cancer. They have an ampullary cancer or intrapancreatic cholangiocarcinoma. And so there's some attrition from that point there. 357 are confirmed to have pancreatic cancer that are registered. And about 55% of those goes on to study. We study this attrition. We try and understand why these patients don't go on to study. And there's a variety of different choices, different reasons there that we're working on. One of the aspects is that the attractiveness of a first-line study based on a variety of different reasons in patients and clinician preference. But regardless, that once they're in the system, we prepare them for their second-line studies. So they get a report, and so they're pretty much even at the time of progression, or even sooner if they so desire, they can go on to second-line study. The next slide, please. Now, so what do we see in this early data? These data are metastatic disease rather than primary disease. We don't have a lot of data on metastatic disease. So essentially, we do see a lot of similarities at a genomic level. There is not as substantial a difference as one might have considered. And that's not surprising considering that many patients, even with resected, develop metastatic disease fairly rapidly. So we're seeing a lot of similarities, but some differences. If we can go to the next slide, please. So you can see on this one, you've got the PCORG data set, which is the darker colored one to the left, which is everything that came from whole genomes from primary resectable. And the bar on the right, the reddy coloured one, is what we see in the metastatic setting. Now the next slide, please. So whilst there are similarities, you can see there are some differences. And there's probably an explanation for some of these differences when you're starting to enrich for things like RNF43, TGF-beta receptor 2, Notch1, etc. And this is likely because we're seeing an enrichment for the classical subtype in patients that go on to study. So even though in metastatic disease we see they're probably approximately equal, if we can go to the next slide, thanks. We're starting to see that, and I'll get to that in a second. What do we see from KRAS? We see, G12D, G12V, G12R. We don't see any G12Cs, unfortunately, even though that's the only druggable component. But of course, there are companies that are coming up, such as Mirati, with studies that can target that. If we analyze those that are KRAS wild type, that's the 6.5% that are residual, we can see there's mutations in EGFR, BRAF, B2, FANG-G, and some gene fusions that are potentially targetable, but they're in a very small minority population. One of the trials that we're doing, we're targeting high mutation burden in combination therapy using an IO and a PARP inhibitor. If we draw a threshold of 4 mutations per megabase, we're starting to see a significant amount of patients, 9.7, almost 10%, that have potential targetability for based on tumor mutation burden. These are not the highest, the ones that we see in microsatellite instability. They only constitute about 1%. But the question in that study is to whether the addition of PARP inhibitor can induce further damage and improve sensitivity to checkpoint blockade. Next slide, please. One of the things that we did is to look at what of the trials that we have, what proportion do we have, of all the patients we have and all the mutations we have, what proportion of trials can we see that are eligible? So we think about 17% of patients have some DNA damage response deficiency based on our hypothesized biomarker, which is used for the analysis of the Primer 001 study. And of course, you can see some of the other groups there, porcupine inhibitor, EGFR inhibitor studies, etc., and the pembrolizumab olaparib study I just mentioned a few minutes ago. So there are sizable numbers of patients that can participate in these studies at the current rate that we're recruiting as well. Next slide, please. Thank you. So if you look at the transcriptome, this is the first 150 cases that went through Precision Panc. We can see if we cluster in 2 different ways. You can cluster into 2 subtypes, and you can also cluster into 4 subtypes. Of course, this is evolving, new subtypes that are arriving from Canada, from others, trying to understand the biology and how these all meld together. It may be a challenge because there's overlapping domains. The other aspect is that there's no clear distinction between the different subtypes. There seems to be a spectrum, and so that will, as we accumulate more data, we'll understand more and more of that. Next slide, please. So this slide shows the proportion of the squamous and the classical subtypes. Those that come into the study, that enter Precision Panc, and those that go onto the study or not fit for study. And you can see that clearly that tracking is the poor prognostic aspect of the squamous or the basal-like tumors that we understand. We can see there's clearly enriching for more the classical subtype for patients going on to study. So that's an important aspect to understand when designing a clinical trial. If you're particularly trying to target the squamous subtype, you can see that if you're targeting the squamous subtype in your study and it's only present in a quarter, you're already underpowering your study by 75%. There's a lot of activity trying to define these transcriptomic subtypes in a diagnostic that will be clinically relevant for recruitment. Certainly there's a number of companies doing that. We're working with a solid tumor TempoSeq platform, and as you saw previously in the clustering, the data is coming out from FFP material, which is quite usable, so watch this space. Now the next slide. And I just wanted to finish with what do we do with these data that we generated in Precision Panc, other platforms that come from all around the world that are generating these data? How can we bring them together and share them in a way that includes rich clinical data and allows people to interrogate these data parcels and understand more about cancer in general and from our personal passion, pancreatic cancer. And this is the new initiative of the International Cancer Genome Consortium called ARGO, Accelerating Research in Genomic Oncology. And our goal is to deliver 1 million patient years of precision oncology knowledge to the world. That exemplifies the importance of longitudinal data. It's not just a snapshot of 1 million patients. It's 200,000 patients with 5 years follow-up, 300,000 patients with 3 1/3 years follow-up. So that gives us an understanding of the clinical endpoints. And ICGC ARGO is now very much about the patient, about the clinical question. Of course, strong, robust genomics and genomic science is the fundamental basis for it, but the questions that ARGO wishes to address are of significant clinical relevance. The next slide, please. And these questions are the sorts of questions we'd like to answer to tell our patients. What sort of cancer do I have? Do I need treatment? Can I access the treatment? And this is all for an individual rather than on average. At the moment we have, it's grown since then, I think we have 17 countries on board. We have about 100,000 promised participants, and there's now up to 30 programs from around the world. And so I'll end with that. If you could do the next slide, please. A lot of people to thank. These are just the key people, and of course there are many participants and supporters all across the UK, some 240 clinicians and scientists part of Precision PANC. Next slide, please. And of course a lot of support from funding bodies and a lot of advice from them as well. Next page. And I think that's it. Thank you very much for listening. And I'm happy to take any questions.
Thank you very much, Andrew. Let me ask you a first question. How is it organized at the national level, these projects?
So the Precision PANC project?
Yeah.
Yeah. So essentially we had to build the system. So there are infrastructure provided by the National Institute of Health to run clinical trials and clinical trials in cancer. So in order for us to do that, we need to bring the community together, come up with a strategy and a plan, design the studies, attract industry investment and industry support. Then once we had approval for the study, we were able to access that national infrastructure for clinical trials and through the NHS. And so the costs we had to pay for was the molecular profiling. We had to pay for any excess treatment costs beyond the standard treatment of patients. And essentially that's how the structure runs, that our industry partner pays for the clinical study and contributes to the molecular profiles. We of course secured grants to start the process rolling in order for us to have staff and develop the processes required. We are pretty close to self-sufficiency based on investment from our industry partners, but we're not quite there yet. We are still somewhat dependent on some grant funding, but it's envisaged within 18 months or 2 years that it will be a self-sustaining program where we are attractive to industry partners to run their clinical trials through our platform. Did I answer your question, Fabien?
I have a very bad connection. Maybe you can relay on the questions, Delphine, or...
Okay, so I'm going to relay Fabien, and maybe do you have questions from the floor? Can you get questions? I have one.
Yes, I have a question from Marseille. Can I ask it?
Malat, or just one before, please. Nuria Malat is asking, do the data you represented represent all PDAC regardless of their stage? How the dynamism of the disease impacts on treatment? Did you hear my question?
Je n'entends rien du tout, mais ça marche.
Si, moi j'ai entendu si, hein.
You broke up a little bit. It was hard to understand.
So the question was, do the data you presented represent all PDAC regardless of their stage?
No, this is the data I presented was metastatic PDAC. Metastatic advanced PDAC recruited into Precision PANC. So it's advanced metastatic.
Okay, and another question was, how often do you have the changes with treatment? Or you have to change the treatment schedule, actually. That was exactly the question. How often do you have to change treatment schedule?
So in the trial itself, so the treatment schedule doesn't change within the trial. The treatment schedule will continue as per protocol. and then at progression or withdrawal, then that's when the endpoint hits. If they wish to, for another therapy, we have second-line therapies they can enter into. So you complete one for your first line, and then you can enter into the second-line studies. We don't have, within the design, there is no room for alteration of treatment.
Okay, thank you. Nelson, do you have a question?
Yeah, thank you very much. I would like a question that we ask ourselves very frequently. What do you think about the intratumoral heterogeneity? Because you will have a biopsy from a part of the tumor. How you will be, to be precise about other parts of the tumor that you don't have the biopsy?
Yeah, so good question. So the critical aspect around this is, what are you trying to target with your therapy? So in other cancer types where there are effective therapies, certainly there'll be areas of the tumor that may have different mutations, may have resistance mutations, and that may or may not be important to know from a drug development perspective. From the viewpoint of pancreatic cancer, we are still drugging truncal mutations. And so if there's a truncal mutation, by definition, it should be present through the entire tumor. We would be fortunate, I would be delighted to have a treatment that works so well that we were in the stage of looking for resistance mutations in different parts of the tumor. But sadly, we're not. So we're still targeting purely truncal events.
Thank you.
Thank you. Any other questions on the attendance? One.
Yes, Andrew, I'm sorry. Do you develop, I've seen that you're developing at the same time experimental models.
Mm-hmm.
Do they fit with your observation
Yes, so
it's,
so this is an interesting question. So there's two sort of models. There's the patient-derived cell lines that we've developed and the PDXs, where we have managed to get alignment between patient treatment and cell line response. In the very few instances that's the case, if we go back, we can see that those lines respond to a particular treatment, if a patient did respond to that particular treatment. If we take the specific mutations that we identify through these sequencing efforts and generate transgenic mice or genetically mouse models of this, if they are purely that particular driver, say on the back of KRAS, you do get to see responses if you're targeting that mechanism. But there's something about the combination of those 3 or 4 major elements, P16, P53, and SMAD4, that contribute to the significant resistance that we see. So if in a pancreas, if you make a P10 null model on the KRAS background and you treat it with everolimus or one of the newer agents or rapamycin, you do get responses. But as soon as you put in all the other mutations in the milieu, you don't see responses anymore. And a lot of interesting information is coming from Drosophila, from fly models. So if you can model in a Drosophila more than 1, 2, or 3 genetic events. You can model as many as 15. And so the more genetic events you add, the more resistance these cancers are. And even if they have a clear target, if you add all these other things that may not be, we don't know whether they're passengers or drivers or a bit unclear, you still get resistance. So there's something about that complexity that we see, particularly of those 3 or 4 combinations. Just KRAS, P53, SMAD4, and P16 is just really, really bad. And we don't know enough about that biology of that combination as to why it's really bad.
And I have a question by Michel Ducreux. He's asking, how do you deal with the... Can you hear me?
Yes, yes.
Yeah. How do you deal with the multiple pathways that can be activated in a given tumor? And when you target one pathway, another deleterious pathway can be activated.
Yes, absolutely. The fundamental issue we have is that we haven't even asked the question, is that if we target the right molecular subtype with this particular drug, will it work? So for example, think of the old drug erlotinib and the PA3 study, where there is a positive signal, a very small incremental benefit. The retrospective analyses of KRAS were done using older technology, so we still haven't asked the fundamental question, is if we target KRAS wild type with, like we do in colon cancer, with an anti-EGFR agent, will we see responses? We may or may not, and of course resistance will develop, but we haven't even asked that question yet. So we need to ask those questions to see whether this holds true. And of course, there will be this whack-a-mole, something else will arise, but we need to get some traction somewhere.
Okay, so I don't see any other question, right? So, yes, oh, there is one.
Hi, Andrew. I'm speaking from Paris, Pierre Cordelier. Have you tried to integrate all those mutations into pathways to make decisions, with all the algorithm that can be defined by other groups in the world? Speaking of, do you have this mutation, the other one, and then go right to the node that might be best targeted? Have you tried that before?
Yes. Yeah, so the challenge is those 4 mutations dominate, and that's something we're working with Ross Kagan on, and really modeling them in flies to look at the biology. And they're really nice models. And I know people say that flies are not a good model for cancer, but they're a great model for biology and understanding biology. So by mapping out these different combinations, the scary component is as soon as you look at more than 5 or 6 or 7 genes together, you've suddenly got multiple small segments. So at the moment, we're looking at, what are the combinations that we have in the more predominant segments, and then understand response and resistance and what's happening with the biology. What is the product of KRAS, P53, SMAD4, but intact P16? What is the product of this with a KDM6A mutation? I mean, these are all very important questions, but we can't model them in mice. We'll be there for thousands of years. So